Hello, this is Sonia Gonzalez, Master Science student in Omics Data Analysis. I would like to talk to you about how we can model omics data to explore the structure of the genome. I choose the paper for Ming et al. Understanding Spatial Organization of Chromosomes via Statistical Analysis of hazy data. I'm sure that you're hearing about the length of the DNA and how it's packed in the nucleus. DNA of our cells is 2 meters long, but it has to be packed inside a nucleus that is 10 micrometers of wide. So, how it's possible? The analysis of data by traditional methods as microscopy in cytogenetics, molecular cytogenetics, or electronic microscopy have limited throughput. No more resolution is possible than the 30 nanometers fiber by these techniques. So what's the next? Wonderful advances are now made thanks to the use of molecular and computational methods that allow us to model in structures or explore the whole genome interactions. The walk from the data to the structure is an amazing and unexplored way. Here we present some of the last advances in this field by the use of techniques as the hazy method. The idea is to figure out which parts of the genome are touching between them when the genome is folded up. Technical procedure starts by cross-linking protein and DNA in place and cutting the genome into tiny pieces. Then the ends of the DNA fragments are marked with biopsy. By ligation, these sequences that previously were closed in the three-dimensional space were now glued together into a single molecule of DNA. Finally, we release the biopsy, and by using high-throughput DNA sequence, we see where are placed these sequences in the genome and how many times or how often they contact. After DNA sequence, two sets are generated, forward and reverse row files. And now we will follow the application of a pipeline for data processing. We need to define the valid reads, so these ones must be unique and mappable. So we truncate the paired ends, removing the nucleotides that were filed in the Janssen position at the ligation step. By this, we prevent interference in alignment. Next step is to map the reads to the reference genome. Then we need to filter to remove some technical artifacts as self-ligation reads, unligated tension reads, random break reads, and PCR duplicated. PCR duplicated are fancy filtering by removing the top 0.5% of the fragments with greatest number of hits, which ones are prone to be PCR reads. Some other fragment-level filtering include to remove low-score mappability reads and thresholding the fragment size. Some bias adjustments are necessary. Some of them are due to properties of the reference genome. For instance, the bias due to the restriction enzyme is due to a non-uniform distribution of the restriction sites among the genome. After selection reads, alignment, filtering, and adjusting the bias, the overall result consists in a count number of co-occurrences between the positions in the genome. This dataset is called the contact matrix. High-C contact matrix is generated at the low resolution level according to the pairwise 3D distance matrix, following an inverse relationship between the three-dimensional distance and the probability of contact. More distance, less probability of contact. The heat map representation of the contact matrix is called the contact map. In this map, each pixel represents all possible interactions along the genome, and the intensity of colors corresponds to the total number of reads where the interaction of one position to other position is observed, as more reads, more intensity. The expected without any interaction will be the plot of a diagonal line where the maximum probability for a position is for itself and for the flanking nucleotides. But in the genome, the observed probabilities for contacts are clustered in discrete regions along the chromosome. If you think that is not easy to understand, we can try a more didactic scheme. Let's imagine that you're watching the Simpsons show. Let's annotate how many times two characters feature in the same scene. By counting medians, we can analyze the proximity between elements. Each one of these clusters is called the contact domain. But well, what about the meaning of the contact domain? When we zoom at the end of the loops, we almost always found a protein called CTCF. CTCF cannot bind just anywhere in the genome. It binds in places with a particular motif writing in the DNA. We found that the same motif is usually found at both ends of the loop, pointing toward one to other. And the same epigenetic modifications are shared inside a loop or domain, defining a genome partition. A principal component analysis for functional features defines this partition of the human genome into two main compartments. Here, the gene vectors are plotted as negative or positive influencing components along the chromosome. Compartment A is associated with gene-rich and actively transcribed regions. Compartment B is associated with gene-poor sequence and reversibly transcribed regions. And here is my personal contribution to this slide. As my cytogeneticist background, I could find a good fit of the Asian vectors with the cytogenetic chromosome binding pattern. It makes sense as our bands are too flavored in functional properties. In the study of biophysical principles of chromatin folding, two models, the fractal globule model and the equilibrium globule model, have been proposed and debated. The read count data from high-C experiments allow us to evaluate the average frequency of chromatin interactions for any given genomic distance. The theoretical predictions using high-C data can be used to fit these two models, providing us experimental evidence of better adjustment of one of them. To study the structural variation of the chromatin using computational approaches called batch-mix, they are tested under the assumption that the genome is organized as a train-like structure. This structure is dynamic by chromatin extrusion in CTCF complexes, and it evolves among the size cycle. More experimental data is needed to validate and refine the actual 3D modeling approaches. So we can remark that high-C technology is important to understand the genome architecture. A great research impact is achieved in functional genomics, DNA replication, cancer, and evolutionary biology, among others. Further efforts will allow to explore the prediction of boundaries in topological domains and the identification of long-range chromosomal interactions. But this is not so simple due to multiple layers of biases, noises, and uncertainties in the high-C protocol that will require novel statistical methods to be developed. And this is amazing because we just can form part of the people that join their efforts with biophysicians and statisticians, resulting in a more meaningful and integrative results. Thank you very much. I hope that you have enjoyed this presentation and don't hesitate to contact for any question. Bye.